Okay, this is the continuation of the lecture on trihybrid crosses. And I strongly recommend, I know I said this at the end of the last one too, but I just want to really emphasize that I think it's a very, very good idea to rewrite the genotypes or the phenotypes, but in the correct order once you figure that out. Because if you don't, it's harder to figure out exactly where the crossovers have taken place, and that is going to be, that could, especially during an exam, it feels wrong to take the time to rewrite them, but if you don't, then you sometimes calculate the distances incorrectly because you don't know where the crossovers took place, or it takes a really long time to figure that out. Okay, so for the two larger classes, we know that no crossing over has taken place. And then when you have, I don't know why that keeps happening, when you have the genes in order, it's really easy for you to know where the crossovers took place. So this category, the crossovers took place between purple and black. And so I recommend just, I like to number them. You don't have to do it that way, but if I, this is just, I'm sharing the technique with you that I generally use. And it is to go, okay, this is region 1 and this is region 2, and we're going to count up the crossovers in each of those regions, right? So this is a single crossover in region 2, and the total there is 85. Right? This is a single crossover too, but it's in the region 1. Okay, that you can see that that's where the crossovers took place, and therefore that is 183, I think. And so we can tell already, without doing the actual mapping, we know that the distance between vestigial and purple is going to be bigger than the distance between purple and black. And then here's our double crossovers, and something is wrong with my slides because they should have both crossovers, right? And so hopefully the color coding and the bolding and so on helps you see that more easily, but you've got now double crossovers, so there's been two a crossover in each of those regions, and there's 8 of those. Okay, and our total is 1448. And I know it takes time to organize the data and to work this part out, but it makes then the calculations more straightforward. So you basically look at the purple to vestigial distance and the purple to black distances. You add up all the crossovers that you could detect between them, divided by the total. That's going to give you the R value, and then we multiply that by 100 to get our map unit, 13.2 map units. And then here's our second interval. It's a shorter interval, so we add up all the crossovers and divide by the total. That's our value of R, which we multiply by 100 to get our map distances. And so this is our full distance. So 13.2 map units between vestigial and purple, 6.4 between purple and black, and then 19.6 by adding the two together. Okay, and the importance of double crossovers can be shown in this case, where if we were to just look at the genotypes and go, okay, well, there's a crossover between vestigial and black here, and here, of course, and here, and here. But if we just look at vestigial and black, we wouldn't include these guys in our calculations, but in fact, we should include them twice because there's been 2 crossovers. So each of these individual counts as 2 crossovers. If we did the calculation by only looking at the 2 outside, vestigial and black, ignoring the purple gene, we would get this distance, and it would be underestimated, right? You'd see that this number is more than a map unit higher than this number. And the reason is because we haven't included those double crossovers. So when we do the calculation, and on an exam, I am likely to ask you, please calculate all the map distances. And in that case, I am looking for you to include these and to count them twice. And that will give you the correct distance, right? And to even, if I say explain, we have to include the double crossovers and we have to count them twice because each individual represents those two crossover events. Okay, so now I've been saying a lot that the probability of crossing over in any one region is the same. And there is a phenomenon that we have observed, which is called interference. And it's not a really strong effect, but it is an effect. And basically interference is when there is either suppression or increasing the number of double crossovers. That means if a crossover occurs in a particular location, it's sometimes more likely that another crossover is detected nearby. That is seen in prokaryotes, and we may talk about that when we talk about bacterial genetics. And in eukaryotes, we more often see that if there is a crossover in one region, it's less likely that another crossover occurs nearby. And when I think about it, I always think about, there's a lot of stuff going on in the chromosome. The DNA strands break, and there's these different proteins that get in there, and crossing over and DNA repair involve some of the same kinds of proteins and processes. And it feels to me, this is just a thought, that the actual proteins that assemble on the DNA to facilitate that crossover, they kind of hog the space so that another crossover event can't really happen nearby. So there is a small amount of suppression of double crossovers. And it's different in different experiments, and so it's just something that we are aware of and we calculate. Okay, and so it may be positive or negative. If interference is positive, that means we've had fewer double crossovers than we might have predicted, and we see that usually in eukaryotes. If it's negative, that would mean there's no interference. There's in fact almost like an encouragement of more double crossovers than expected. This is seen in prokaryotes, and I think the reason for this is because those chromosomes are circular. So if you have one crossover under the kinds of ways that we detect them, you sometimes can't recover the organism because the chromosome has been made linear instead of a circle. So you sometimes need two crossovers to keep the circle intact. In eukaryotes, it's more of a fuzzy concept. Okay, so, but anyway, the third step, after we've made our map, we want to calculate the coefficient of coincidence. This means the number of crossovers we would have predicted based on the map distances we calculated, and then how many we saw. Okay, and the coefficient of coincidence is the number we saw divided by the number we expected. If we got exactly what we expected, then the coefficient of coincidence is going to be 1. But if we saw fewer than we expected, then it's going to be below 1. Okay, and if we didn't see any, then the number would be 0. Right? So you take the expected number of double crossovers, the probability of crossover in the first region for our experiment, 0.132, multiplied by the probability of crossover in the second region. That's what we're detecting. The crossovers we are detecting, there's one in one region and one in the other. If there were two in this region, we would not be able to observe those. So of the ones that we could detect, how many should we have seen? And if you multiply that by the total, you will get 12 as your number. But we only saw 8 in our experiment, so our coefficient of coincidence is that 8 divided by 12, which is 2/3, so 0.67, right? So, and interference is 1 minus that. So interference is 0.33 or 33%. And what that means is that tells you how many of the double crossovers that we would have expected to see were actually suppressed, were not actually seen. And that's what that means. Okay? And I'll probably have some questions on this in the actual class, just to try to hammer away at the topic. It can be confusing. So we can map a lot of different genes relative to one another, and before even the genome projects came out, we now have the mostly full genome of most of our model organisms. There's certain regions that are a little hard to get at. But basically, you can map a lot of genes with respect to each other. And if you think about chromosomes and stuff, a lot of chromosomes in flies and other organisms, if you were to map a lot of genes along them and you added up all the map units, you'd get like 140 map units, right? Or 200 map units. Oops, not nanograms. What am I doing? Map units. And so you don't do this across the whole chromosome, but what you do is you map little regions, and then you map some other regions that overlap that, and then you map other ones that overlap with that. So I've done, 3 genes here, and I've done 3 genes here, and I've done 3 genes here, and maybe I've done, I've mapped 3 genes here, and so on. And the idea is you take all the little separate experiments where we really like to map small distances because they're the most accurate, but then you put them all together, and if these 2 genes are linked and these 2 genes are linked, then we know all of this is linked. And if this gene is linked to this gene, then we can extend this whole thing a little bit further. And if these 2 genes are shown to be linked to this gene in another experiment, then we can extend our map further and further. So really it's like every little map is a little puzzle piece, and then you just connect them together as well as you can to get the most comprehensive map. And we refer to these as linkage groups. And at the beginning, when people were doing a lot of this kind of mapping work, they would talk about linkage groups. And now we know that each linkage group, once they became complete and all the regions had been properly mapped, each linkage group corresponds to a chromosome, a full chromosome. And this is obviously not something for you to memorize, but this just shows you some of the work done with fruit flies a long time ago. It just shows you like the length of the chromosome and some of the genes that are found on here. Here's the white gene that I've been talking about, and we'll talk about more. Yeah, black body here, purple eyes, vestigial wings. So this is a section of that chromosome, that we've talked about those. And so this is, just for your interest, it shows you this is the results of lots and lots of different experiments that have been done to put together the map. And of course, these are not the only genes that are known on these chromosomes. There's lots more all over the chromosomes. The fruit fly has about 14,000 genes. So this is just a very small sample of them. Okay, so the map distance, when you're actually doing maps like this, linkage maps, it's most accurate when you have genes that are really close together, because if you have genes that are really far apart, say 10 map units apart, then there's a 0.1 times 0.1% or chance, or 0.01. So there's a 1% chance of getting 2 crossovers between them. If you've got 40 map units, there's a 16% chance of finding, because it's 0.4 times 0.4, 0.16, 16%. There's a greater chance of getting double crossovers. So what we want in general is we want to keep our map distances that we work on in around the 10 or lower map unit range, because then we can feel more confident that we're not missing a lot of the crossovers based on those double crossovers that give us back parental allele combinations, and they look like no crossovers taking place, even though two crossovers took place, right? So we prefer to work with smaller map distances and then add up the distances over the long term. And the larger your map distances get, the less accurate they're going to be. And this is just in theory. So in theory, in theory, for any two genes, that we should see this kind of linear relationship between the crossovers that we can observe and the map distances. But what we actually see is this kind of more curved graph because of, the larger the map distances are, the more the double crossovers occur, and that means we're just not able to detect those crossovers. And so we'd like to keep the map distances that we're calculating and that we're working with down in this range, because we know this is the most accurate and the most reliable. That when we actually detect, something that's, say, 6 or 7 map units apart, we know that that's actually really close to the truth. Whereas if we're even looking at something that's only 20 map units apart, we're still not seeing, we'll calculate it incorrectly. We'll say it's really 15 or something because we will have missed, that small-ish, but a proportion of double crossovers. So keeping it accurate means map the smaller ones and then just add those together to get your whole larger distance. Okay, so this is basically a summary. First of all, for this first section that I've covered, how to map 3 genes, how to go about the actual practice of solving the problem, and then a little bit of theory about, why larger distances are mostly an underestimate. They're really bigger than we think they are, and how in practice it's better to map shorter distances and then kind of add those up. Where I want to go next is to go beyond 3 linked genes, which we've just done, and talk about the patterns we would see in the cases where, what if we're doing a trihybrid cross and the genes aren't all linked? What if 2 are linked but 1 is on a different chromosome? What if all 3 are assorting independently? What kind of patterns will we see in that case? If they're all linked, we're going to see these 2 really large categories, these 4 in-between categories, and 2 that are very small, and we know that's because you have the non-crossover ones, a bunch of single crossover ones, and then a couple of double crossover ones. And that's a diagnostic pattern. Scientists are always looking for patterns, right? Especially in genetics. And so being able to recognize those patterns is part of the work of learning genetics. So think about, what would you expect to see if you had, 2 genes that were on one chromosome, but the other gene in the trihybrid was actually on a different chromosome? What kind of pattern would you expect to see? And then what if all 3 were on completely different chromosomes? What would you expect to see there? And that's where we'll start up next time.